morning, good afternoon, good evening, and welcome everyone to Global IMMUNOTALK's 2022 today's seminar. My name is Burkhard Becher, and on behalf of the other organizers, I welcome you all, and I welcome our guest for today. That's Adrian Liston, and I'm very excited to have him here. And I would like to share with you, so I'm glad that you could make it to Global IMMUNOTALKs today. As you know, at the end, you will have free access also to the seminar via YouTube. You just have to look for it and search on YouTube for it. I'm very excited to be part of this initiative because it allows really a lot of people to hear seminars that we don't usually hear. It's very an inclusive way of sharing cutting edge seminars worldwide with a very small carbon footprint. At the end of the seminar, Adrian will answer questions via honor. Before I say that, maybe this is the speakers lineup for this year that we have planned. So today we have Adrian Liston. Next week is Frank Quintana. I urge you to listen to all of these speakers here. It's an easy way of doing it. You can do it from the comfort of your home. And let me stop sharing this right now. It's such a pleasure to have Adrian Liston with us today here, who is, I have to say, one of my favorite immunologists that I know, and I'm particularly pleased that he could make it here today. And Adrian is a senior group leader at the Behavioural Institute, and he's a senior fellow at the Churchill College in Cambridge, in the United Kingdom. Adrian is an Australian. He did his PhD with Chris Goodnow at the Australian National University, and then he did his postdoc with Sasha Rudensky when he was still at the University of Washington. And I got to know Adrian at his time at the Vlanders Institute for Biotechnology at the VIB at the University of Leuven in Belgium, where he basically started his lab and did some just amazing stuff there. And then in 2019, I think he wanted, because it coincided with Brexit, he thought maybe that was attractive and moved over to Cambridge. So the laboratory works, as many of you probably know, on understanding the interaction between the immune system and tissues. In particular, lately, much of the work he's done was really fascinating work on the brain. Adrian is in spite of his young age, he is a fellow of the Academy of Medical Sciences. He won the Eppendorf Prize. He's a Wellcome Trust investigator. And among many other things, he already holds 3 ERC grants. So Adrian, super pleased and super excited to have you here. And the one thing I forgot to mention, you happen to be one of my favorite speakers. So I will very much look forward to listen to you. Now, before we get started, there's one thing that you may have heard that we do with Global IMMUNOTALKs. We like to ask a question, a general question that may be of interest to others. And so in a few short sentences, maybe can you answer the question, what would you do differently if you were to start your lab again?
Yeah, thank you very much for such a kind introduction. I think it's really important to recognize that making mistakes is part of the process of starting up a lab. I would say the main thing is to remember, if you fail fast and then correct your course. Mistakes are the process. As long as you make a mistake and then correct it, then you're fine. For me, I would say that probably one of the hardest things to get right is the appropriate level of investment in your team. For people who tend to overinvest, like me, that meant learning that sometimes the best strategy is to end a position, such as when you have a toxic staff member. While for people who tend to underinvest, that means they really need to learn that their staff also need time and space to make mistakes and improve. But it is such a hard thing to get that balance right.
I agree. These are wise words. I just had a discussion the other day with a young investigator who basically had the same. I'm sure she'll hear you today, and we'll take this advice. All right, super. So I'm sorry I pushed this along. Let me just make sure we get this started. So if you could start sharing your screen, Adrian, we're eager to hear what you want to share today. Very good.
Excellent. Thank you very much. So today I'm going to be talking about neuroimmunology, and in particular about manipulating the population of brain-resident regulatory T cells. So the brain is historically thought to be a site of immune privilege and rather isolated from the rest of the peripheral immune system. However, this has really been changing conceptually over the last few years, led in particular by investigators such as Kipnis and Schwartz, demonstrating that there are neurological changes in various immune knockout mouse strains, characterization of brain lymphatic drainage, and also discovery of direct vascular channels that link the skull bone marrow to the brain. So the idea that the brain is isolated apart from the immune system, I think, is slowly being challenged and being replaced with the idea that the brain has a special relationship with the immune system, but it does have a relationship. Now, for us, we came to this question by looking at whether or not T cells are existing in the healthy brain. And this was rather controversial. There were reports of T cells found in the CSF and even in the rodent brain, but I would say it was still not widely accepted that there are CD4 positive T cells present in the healthy brain. And the main issue is that reports were rather incomplete, and it was very easy to dismiss reports as being due to contaminating blood cells, which is a real problem when you're dealing with such a rare population as T cells present in the brain. So I'm going to tell this story in two parts. The first part is more of a developmental story about CD4 T cells in the brain and the development of the brain. So first of all, what are these CD4 T cells in the brain? And here I just want to show a few images to get started by James Dooley, our extraordinary histologist in the lab. And this is an example of a T cell in the mouse brain that is just in the process of crossing over from the leptomeninges into the brain parenchyma. So here you can see there's a double laminin barrier, and this CD4 T cell in green has stuck a pseudopod beyond the laminin 4 barrier, but it's still inside that alpha 1 barrier. Now we can look at an example of a T cell that has gone a bit further. And here, if we look at the T cell at the bottom in green, this cell is almost entirely beyond the double laminin barrier, with only a few remnants of the cytoplasm now stuck between the laminin 4 and laminin alpha 1 barrier. So this is a T cell that really looks like it's on its way into the brain. And then you'll have CD4 T cells that are clearly in the parenchyma of the brain. So now we're co-staining with GFAP so that we can look at astrocytes, and we can see this is a T cell that is beyond the glial limitans and inside the parenchyma of the brain. So we're able to go through the entire brain, section by section, and really count up how many T cells there are present inside the healthy rodent brain. And we can do this and come up with a number of about 10,000 T cells per gram of mouse brain. Keep in mind that a mouse brain is substantially less than a gram. So it is more like 4,000 or 5,000 T cells total per mouse brain. And a lot of these cells are adjacent to the vasculature, but quite a few of these cells are actually quite distal. So we'll interpret those as being more parenchymal cells. And here it's notable that there are CD4 T cells more or less across every region of the mouse brain if you look, but again, in very low numbers in a healthy situation. The other thing we can do is use flow cytometry to look at T cells present in the mouse brain using a technology that gives us more parameters to play with. So here it's quite important, though, that we use a label to distinguish between those cells that are really inside the brain and those cells that are in the vasculature. So here what we do is we inject the mice with a PE conjugated CD45 just before takedown, and then we perfuse the mouse. So if we look at a mouse that has been perfused and injected with CD45 PE, we see that 90% of the cells that we get out of the perfused brain are genuine brain resident cells, and a fraction of those cells are just trapped in the vasculature at the point of takedown, as unlike the blood control. Now, if we do the same approach, we see, again, about 10,000 T cells per gram in the mouse brain. Again, we see the numbers more or less spread across the different regions. Here I would point out that in terms of absolute numbers, there are more cells, more T cells present in an area like the cortex than the meninges. Of course, the meninges is much more enriched in CD4 T cells, but it's such a small area that if you take the total number, the cortex has a higher number than the meninges. And importantly, if you use flow cytometry or imaging, you're getting about the same number of CD4 T cells present in the mouse brain. The other thing that we can do now we're using flow cytometry is to look at the markers that are present in the mouse brain. So here we're doing high dimensional flow cytometry, so we can compare multiple different markers. We present this as a T-SNE plot for dimensionality reduction. So you can see the different regions and different colors on the plot basically correspond to different combinations of markers. The most important marker, though, really would be CD69, which if we compare the expression on the blood in red or the brain in black, you see a large population of CD69 positive cells in the brain. This population is not present in the blood. The same marker holds true in regulatory T cells. And it's another way of showing that, first of all, these are not contaminating blood cells because they have a different phenotype. And secondly, these cells have got some type of adaptation to the brain. Now, I'll just point out quickly that I have a p-value here comparing the two different T-SNE plots. And this actually took a lot of work, a year of developing a new statistical test that we have called a cross-entropy test. And essentially a cross-entropy test allows you to compare two different dimensionality plots, dimensionality reduced plots, whether or not it's a T-SNE or a UMAP. And you can say whether or not those two plots are statistically different. And if you have more than two plots, you can say which of those plots are closer to each other. So you can really start to do statistical analysis of high dimensional data using a T-SNE or a UMAP. If you're interested in using this technique, you can go to our website. We've got the cross-entropy test. There's a link to our archive paper, the GitHub account, and also YouTube videos for walking immunologists who might be allergic to programming through the steps required in order to use this test. Now, moving from mouse to human, we're also able to look at the presence of CD4 T cells in healthy human brain. Although I say healthy human brain, what we are able to use in this case is the biopsies taken during surgery for epilepsy. And so we are looking at the healthy part of a patient with epilepsy. And if we do this, we get again about 10,000 T cells per gram in the human brain. So very similar numbers to what we see in the mouse brain. And we see, again, a unique signature when we compare the blood cells to the brain T cells. And again, we're getting the same types of markers come up in mouse and humans, of CD69 really being a key marker for those brain resident cells as opposed to the blood resident cells. So what are these T cells doing in the brain? Here we took a clue from some data from James on the location of these CD4 T cells. So this is now back to the mouse. And here we are imaging a CD4 T cell in green, along with microglia in orange. And you can see that this CD4 T cell is really wrapped around by a microglia with a very intimate contact. And this made us ask, well, what effect does the presence of CD4 T cells in the brain have on microglia? So here we used the MHC class 2 knockout mice. So because of the role of MHC class 2 in the pharmic development of T cells, the class 2 knockout mice essentially have no or very few CD4 T cells. So it's like having a knockout of CD4 T cells. If we compare the wild type versus class 2 knockout mice by single cell sequencing of microglia, what we can see on a T-SNE representation is a very clear division between the transcriptional profiles of either the wild type cells or the knockout cells. And the knockout cells are really concentrated in this region of the plot over here, which corresponds to a microglia 3 region. And if we just compare a wild type microglia 3 to the rest of the microglia, we see that there are a number of genes that are downregulated on microglia 3. Although I would say it's actually going to be more correct to say they're upregulated on all the other microglia. And this is because if we look at this battery of genes, we see that if we look across wild type mice and microglia from wild type mice, we see that these genes are generally very low in the prenatal microglia, and they're turned on in the early postnatal and juvenile period. So these are genes that are really not present in fetal microglia, and they're turned on in adult microglia, which means that in the knockout mouse, where we don't have CD4 T cells, these microglia look like they're suspended in that more fetal state without fully progressing to the adult microglial differentiation state. We can also visualize this as a pseudo-time plot where wild-type microglia are progressing through developmental pathways, while the MHC class II knockout microglia, which are growing up without the presence of CD4 T cells, these cells that really seem to be stuck at that early developmental stage and are not following through. I won't go through all the data, but just to mention that we have validated this system using systems that are independent of MHC class II knockout as a genetic manipulation. We can do this by taking mice and injecting CD4 depleting antibodies. If we do this, we block that maturational signature, and the adult mice still have microglia stuck in the more juvenile state. We can also take microglial slices from a neonatal mouse and then add CD4 T cells and promote this differentiation step. Or we can take conditioned media from T cells and use it to directly induce this differentiation signature in cultured neonatal microglial cells. Now, why is it important that these microglia are suspended at the fetal stage in the absence of CD4 T cells? Well, this is because microglia actually gain a new function when they differentiate into the final adult state. And the fetal stage, a microglia has got a lot of function in cleaning up after the development of neurons. However, in the post-fetal state, the microglia are having a new function, which is to enforce synaptic pruning. This is because in the fetal state, there are far too many neuronal connections, many of these very weak. And in order to create good, strong neuronal connections, those connections are flagged, the immature connections are flagged for synaptic pruning. The microglia come on and basically eat off those immature connections. So we can directly measure the number of synaptic connections in the wild-type mouse and the class II knockout mouse. And we can see there's no difference in the number of mature connections, but the number of immature connections is much higher in the knockout mouse, implying that the knockout, without CD4 T cells around, has poor synaptic pruning of these immature synapses that really need to be removed. Now, in terms of how defective this is, we end up having an excess of about 20% too many spines in a knockout mouse. Now, 20% doesn't sound like a lot to an immunologist, but from a neuroscience point of view, an increase of 20% is associated with diseases such as Fragile X syndrome and Rett, while a decrease of 20% is associated with syndromes such as Down syndrome, autism spectrum disorder, and schizophrenia. So we are really talking about a change in the brain programming that can give quite large behavioural impacts. And in fact, when we went and measured the behavioural defects, we were able to show that the mice that are MHC class II knockout, so that don't have CD4 T cells, had consistent behavioural difficulties that implied that they had a difficulty in learning. For example, in the Morris-Water maze, which tests cognitive spatial learning, these mice were consistently slightly defective. So just to conclude part one, we have the seeding of the brain by T cells from the thymus. So these are going to develop in the thymus and migrate into the brain. Once inside the brain, there are some type of factors that are given from the T cell to the microglia. We don't know the exact nature of these, but they do seem to be secreted factors. And these factors are important for the microglia to reach that final adult stage of being able to undergo synaptic pruning. And this is really quite critical for fully mature cognitive capacity. Now, we know that these cells are present in both the mouse and human brain, and that the absence of T cells in the brain in a mouse causes cognitive difficulties. Could you have the same effect in humans? Here we don't know, but there are parallels. For example, if you look at individuals undergoing chemotherapy, they'll often complain of chemo brain, where they are forgetting short-term memory, and you've got HIV-associated dementia. In both of these cases, you've lost CD4 T cells, and you have these neuronal effects. Now, this, of course, could be due to side effects of chemotherapy or side effects of HIV treatment, but it's interesting. And the other point that I think is really quite interesting is if you take children that have a severe combined primary immunodeficiency, where they do not have T cells developing. So this is quite analogous to an MHC class 2 knockout mouse. These children often have a cognitive defect of about 20-point lower IQ than their siblings. Now, again, this could be correlative. These children are spending a lot of time in hospitals rather than in schools, but it is very suggestive of some type of parallel function in humans that I think is worth following up. Now, to move on to part 2 of my talk, here I really want to talk about some exciting work we've done in collaboration with the Matthew Holt lab in Leuven in Belgium. And here we're talking about, can we change the resilience of the brain by enriching the regulatory T cell population? This work came about after my personal encounter with traumatic brain injury 11 or so years ago, when my brother, Russell, died after traumatic brain injury. It was really striking to me how little could be done in this situation. He had a traumatic brain injury, and at this point, it's essentially emergency care. We don't know enough about traumatic brain injury or how to treat it in order to come up with really clever therapeutics. And unfortunately, Russell's case is not rare. Every year, there are 50 million traumatic brain injuries, 5 million hospitalizations, 1 million deaths, and 25 million people are left with a long-term cognitive impairment. This is as big as every cancer put together, and yet it's so under-researched. For 50% lifetime of risk of TBI, we really should be able to do something for patients in this case. And when I was thinking about this 11 years ago, while my brother was in a coma, essentially I thought this should be an easy problem. Inflammation from tissue injury, it is predictable and tractable. If you have an injury in your ankle, you twist your ankle, we know the inflammation, the swelling, the redness, the pain, the warmth. We know how to reduce that. Anti-inflammatories will reduce that inflammation and prevent the long-term damage. And the same fundamental process should be occurring in the brain. It is just a tissue injury. Of course, while this might be an easy problem in the ankle, in the brain, it's an easy problem in a very hard place. There's a few different reasons for this. One is that swelling in the ankle is no problem. Swelling in the brain is going to cause you compression death. Essentially, that you get the initial injury, and then you get this secondary neuronal death because of the swelling against the brain, of the brain against the skull. And then you've got the additional problem of the blood-brain barrier, which prevents anti-inflammatories, classical anti-inflammatories, from getting to the brain to reduce this inflammation. And you can have a chronic inflammation that really lasts years after the initial insult to the brain. And this made us wonder, why do we have this chronic inflammation? An inflammatory event after a swollen ankle goes down by itself after a week. In the brain, you can really get this swelling and this inflammation occurring for a year. And our thoughts were that maybe the reason is that the regulatory T cell numbers are just too low in the brain to stop that chronic inflammation and turn it into an acute issue. So could we prevent the chronic inflammation if the Treg numbers in the brain were just higher? So first we had to work out, why are Treg numbers in the brain so low? We did a parabolic experiment where we joined the circulatory systems of CD45.1, CD45.2 mice. You can see them as red and blue mice. And essentially what you do is you join that circulatory system. So now you have a common interflow of blood. So all blood cells are now a mixture of red and blue, but they're going to go into red or blue tissues. So we can go into the mouse and look at every different tissue for the normalization rates. And so if we go into the liver, for example, we can look at the host Tregs that are entering, the donor Tregs that are entering, displacing the host Tregs. And what we see is that it's really the activated Tregs that enter, and 95% of those Tregs are leaving or dying within a day. Very high death rates and exit rates in that first day. And it's only really 5% of the Tregs that enter that are converting into a more residential population. Here we define that as CD69 positive. Now, the cells that do convert into a CD69 positive residential Treg, these cells do stick around for quite a long time. Eventually, half of them will die, and the other half will leave after an average of about 2 weeks. So it's a small number of cells that are able to seed the tissue, and they last a number of weeks, and then they're being replaced by incoming cells. Here, we're able to do some comparison now across multiple different tissues. And we use Markov chain modeling, which can calculate the population flow by taking the starting state and predicting what happens next. Just as a reference, this is more or less the same system that autocorrect on your phone uses to try to predict what word you're writing. So if we use this Markov chain analysis, we can calculate from the parabolic data what is the entry rate of regulatory T cells into different tissues. And so you'll see lymphoid tissues have a very high entry rate on the left. The non-lymphoid tissues have quite a low entry rate when we model this, but the brain is no different from any of the other tissues. So these activated Tregs are able to enter the brain at a normal rate. What happens though is that the death rate is much, much higher. So in general, regulatory T cells have quite a low death rate if they're living in lymphoid tissues. To me, this makes a lot of sense. The lymphoid tissues, like the spleen, have really evolved to host lymphocytes. So they create a great environment for the lymphocytes. Our tissues have got different functions. And when we go to the non-lymphoid tissues, we see that the activated Tregs that are in these tissues have a much higher death rate, 10 to 100-fold higher than they do in lymphoid tissues. And among these tissues, the brain really stands out as having by far the highest death rate of the activated T cells. And so this tells us that the reason why regulatory T cells are so low in the brain is not that they can't get in, but they tend to die once they're there. So that could really be a key intervention point. Fortunately, we know quite a lot about what controls the death of regulatory T cells. This is just summarizing some work from my first PhD student quite some time ago, nearly 10 years. And what he found is essentially that regulatory T cells are pre-programmed to die. These are cells that are always on the edge of dying. And this is because they have a high expression of BIM and it induces the back-to-back apoptotic pathway. In order for these cells to survive, they constantly need to have interleukin-2 provided, and that interleukin-2 upregulates an anti-apoptotic gene, MCL1, allowing the cells to survive a little bit longer. So they are pre-programmed to survive. They're pre-programmed to die, and they just need IL-2 to survive. If we look at IL-2 levels, we can see IL-2 levels pretty decent in the serum and at lower levels in other tissues, but they're really extraordinarily low levels in the brain, by far the lowest level of any of the tissues that we looked at, indicating that the cells that are entering the brain are undergoing essentially what we can think of as IL-2 starvation, and that this might be why most of the cells that are entering are rapidly dying. So what we did next is we thought, can we come up with a solution to deliver interleukin-2 to the brain and rescue that deficit so the regulatory T cells are able to survive? Here, we're really lucky to work with the Holtz lab and come up with a gene delivery system where we can modify an adeno-associated viral vector, or AAV, so that we can produce interleukin-2 in the brain. This system uses a capsid that is neurotrophic, so you can inject the capsid into the blood, and then it will cross over the blood-brain barrier itself. And then what we did is, rather than use a normal strong viral promoter, we use the endogenous GFAP promoter, which is expressed by astrocytes. And this is really important because even when you get off-target transduction by the AAV, you don't get any IL-2 production in the wrong sites, because it's really only the astrocytes that have got the right transcription factors to activate the GFAP promoter and to drive IL-2 production. And we can look here at the large increases we can see in regulatory T cells when we give different doses of this AAV IL-2 delivery. This is all occurring in the brain. In the periphery, we never see any extra IL-2 production. We never see any extra regulatory T cells. Now, I'm just going to be talking now about this 10 to the 9 dose, the lowest dose we looked at. Under this dose, we're increasing IL-2 levels from under 1 picogram per mil to about 3 picograms per mil. And so for reference point, this is essentially bringing the level of IL-2 found in the brain up to the level that we find in the serum. We're not overexpressing IL-2 compared to the endogenous level. We're just rescuing that relative deficit that's present in the brain compared to the serum. And when we do this, regulatory T cells increase in number about 10-fold. And for the first experiments I'll show, we injected the mice 14 days prior to testing. Now, one thing I'd like to mention here is that the GFAP promoter doesn't just target astrocytes. It's particularly strong in what are called reactive astrocytes. And these are astrocytes present in the area around inflammation. So if we look at a healthy hemisphere of a mouse with traumatic brain injury, we can see a couple of green cells here. These are the cells that are expressing our cargo, rare cells. Now, in the same mouse, but the other hemisphere, we have a traumatic brain injury, and you can see a lot more cells that are delivering the cargo and delivering the cargo to a much higher level. So even though both hemispheres of the brain got given the same dose of our AAV delivery system, the production of our biologic, our IL-2, is really concentrated in that inflammatory zone around the injury because the GFAP promoter is upregulated in the reactive astrocytes. So how does this work in a traumatic brain injury setting? What we did is to use a controlled cortical impact model. This is a preclinical model where we remove the skull, we create an impact on the brain, then we put the skull back on the mouse. Over the next 2 weeks, the brain swells. The swelling creates this compression injury against the surface of the skull. And you can see here that at the impact site, we have essentially lost a lot of neuronal tissue in a control mouse. This is just a surface tomography, but I think it's quite obvious, even if you just look at the surface of the mouse, that you have lost a lot of neurons 2 weeks after the injury. Now, if we treat the mouse, and this is given 2 weeks before the traumatic injury, we can see that this mouse has now got more regulatory T cells present in the brain. It still has an impact site. You can still see the scar where we created the impact. What we don't have is this massive crater where we've lost all of this neuronal tissue. We can also measure this by using immunofluorescence. This is, on the left, a nice, healthy, thick cortex. On the same mouse, on the right, we have a mouse that's given a traumatic brain injury. And you can see that you've really lost all of the cortex because the midbrain has swollen up after the injury and just squeezed this cortex to death. If we look at our treated mouse, again, we have the healthy hemisphere and then the damaged hemisphere on the right. We do see signs of damage. We certainly see loss of tissue, but we do see a layer of preserved cortical tissue that's left. And essentially the lesion size, whether or not we measure it by histology or by MRI, is essentially halved if we've got a mouse that has got extra regulatory T cells present to reduce that inflammatory response. It's really important for us also to look at the cognitive outcomes. So here we use a Morris-Water maze. In the Morris-Water maze, you put the mouse in the maze. It's essentially a dish of water. They don't like swimming, so they swim around to try to find a platform where they can get out of the water. If you do this 5 days in a row, the mice remember where the platform is, and they are faster at swimming over to the right part of the dish, and you can time how long it takes them to get to the maze. Essentially, healthy mice learn over this course of 5 days, while a traumatic brain injury mouse really has very much impeded learning. And a traumatic brain injury mouse, given our IL-2 treatment, learns as good as an uninjured mouse. So here you can look after 5 days. The healthy mouse, very slow learning in a traumatic brain injury mouse, while a traumatic brain injury mouse treated with IL-2 is, if anything, even better than a healthy mouse. We've done a little bit of work to try to work out what is the molecular mechanism by which the IL-2 is going to protect the mouse. Because even though we expand up regulatory T cells tenfold, they are still relatively rare cells in the mouse brain at this point. So we think that they're likely to be acting through an intermediate population, such as microglia. Here we've done single cell sequencing, and we have our sham mouse treated with IL-2, our TBI mouse, and our TBI mouse treated with IL-2. And this is just dimensionality reduction on the single cell sequencing for the microglia. And essentially what you see is the IL-2 induces this population of MHC class 2 high microglia. The injury induces this population that are known as disease-associated microglia, or DAM, while the treated mouse with TBI has both of these populations present. Now, if we look at the populations, the markers that are changing with IL-2 treatment in TBI, we see this upregulation of MHC class 2 genes, CD74, and SPP1, which is osteopontin, are all upregulated in these microglia that have been treated with IL-2 during a TBI. And I think the expression of MHC class 2 was quite interesting, because if we look here at the impact site, on left we have a control impact site, and on the right we have an impact site on a mouse that's treated with IL-2. And here you can see the edge of the tissue, which is the edge of the impact. And these MHC class 2 high microglia really seem to be lining the edge of this impact. And if they're expressing anti-inflammatory genes such as osteopontin, they might be creating a buffer that prevents the spread of the inflammation further into the tissue. I just very quickly mention that this is a process which should translate into other neuroinflammatory conditions, because traumatic brain injury, regulatory T cells are not just evolved to prevent injuries, they're also evolved to prevent diseases that are autoimmune in nature. And so here we can look at models of multiple sclerosis, such as experimental autoimmune encephalomyelitis. In a normal situation, you're creating an autoimmune response against a central nervous system antigen, and you get ascending paralysis. So the mice will get a limp tail, a paralyzed tail, start to get paralyzed, forelegs, and plateau. Whereas our mice that have been treated, they're going to get a limp tail, and then essentially they start to get a lot better. And so the total cumulative impact is reduced. We've also looked at two different stroke models. The first is a photothrombotic lesion model, which we can activate with a green laser. And we can look at the lesion size in our control treatment or our IL-2 treatment. And you can see, just looking at the lesion size straight away, the treatments are smaller in IL-2 treated mice, again, about halved by MRI. Or we can go to a surgical model of distal middle cerebral artery occlusion. And again, you can see in the control stroke versus the IL-2 treated stroke, and it's reduced in size by about 50% from an MRI quantification. So this does look like it's got an effect much broader than traumatic brain injury. I also just thought I'd show you some very recent data that I think is quite interesting, which is the effect of regulatory T cells may not be important just for neuroinflammation, but they may also be important for cognitive decline in aging. So here what we did is we waited until mice were 22 months of age, which is quite old. We're talking about the equivalent of probably a 70-year-old human if you want to put mice into age years. And we then treat the mice at this age and wait 2 months. So now we're looking at the mice at 2 years of age. We can put the mice into a Morris water maze to try to measure their cognitive capacity. There's actually two phenotypes that happen in the Morris water maze. The first is that the mice are swimming much slower. So on the top, we have these 2-year-old control mice. Then down below, we have on the top, we have 4-month-old control mice. And then the red down below are 2-year-old control mice. So they're swimming much slower because they're old mice. old bones, old muscles. And if we treat our 2-year-old mice with IL-2, we don't have any change in this. These are still old mice that are swimming slowly. However, if you look at their ability to form spatial memories, here you can look at the difference between day 5 and day 10 as to what percentage of time they spend in the correct quadrant. So here they're swimming around, either fast or slow, depending on if they're an old or a young mouse. And some mice are going to spend a lot of time in the right quadrant because they can remember where it is. And other mice will spend just the random 25% of time in the right quadrant. And so if we look, the dashed line is at 25%. So that is more or less just a random guess of the mouse where it should be going. And you can see that on day 10, you've really got a big improvement in the young mice, while the old mice really do not improve particularly at all. They have this cognitive impairment in spatial learning. Our old mice, however, treated with IL-2, they have a cognitive capacity that is indistinguishable from that of the young mice that have been untreated. In terms of how this can be working, we turn again to single-cell sequencing of microglia. And what we can calculate is the transcriptional program that occurs in age. So this is a complex plot, basically a two-dimensional volcano plot of genetic changes transcriptional changes occurring in microglia with age. And essentially, what we're looking at are changes that are present with age in a control situation. So these are genes that are in this side of the plot. On the left side of the plot, they're genes that are down with age. On the right, genes that are up with age. And then we can see what is happening with the effect of IL-2 treatment in age. And so if IL-2 treatment was doing nothing, you would really expect to see the dots form along this diagonal line. Instead, we have a R value of about 0.47, which means that 50% of the changes that are occurring with age can be reduced by the presence of IL-2. And if we look at what is changing with age, these are the key pathways that are altered by aging. So these are the pathways that old microglia have different to young microglia. And so blue is normalized to being closer to aged control. So these are the pathways, a lot of pathways you might recognize, mTOR responsive to rapamycin, longevity pathways, cellular senescence, proteasome, mitophagy, very much what you'd expect in an aging situation. And then this is what happens if you treat the mouse with IL-2. And suddenly, these old IL-2 mice really have the pathway expression that are much closer to that of the young control mouse. So it looks like we're reverting a lot of these age-induced changes in the microglia by having extra IL-2 present in the brain. Now, we're quite interested in seeing if we can go from mouse to human with this system. But there are, of course, differences between a system that you design to work in a mouse versus a system that you want to go into for the clinic. So here we've done some development of a system that gives extra features that are useful for the clinic. We still use the same basic system. We still use the same basic system of having a neurotrophic capsid and astrocyte-specific expression, but rather than directly driving the expression of IL-2 by the astrocyte promoter, instead we are using a response, basically a tetracycline response system that's been modified to respond to minocycline. This is very useful because minocycline is able to cross the blood-brain barrier and is clinically approved for TBI patients. So essentially what should happen is the GFAP promoter activates the tetracycline activator, which in the presence of minocycline can then activate IL-2. So this is a schematic of how the system will work. We can look at the AAV. We've encoded all of the information in the DNA that is delivered by the AAV. You've got this inflammation response element, which is GFAP, and then you've got the safety switch, which is the tetracycline response element. So if we look at a mouse brain, that you can see this expression of this safety switch that's present, and after traumatic injury, because of the reactive astrocytes, it really goes up in the region directly under the traumatic injury. So we're now increasing the expression of the inducer. Minocycline is able to bind, and then the inducer will bind the safety switch response. And that can allow the production of IL-2. So by adding minocycline, we can now get IL-2 being produced in the brain, and in particular, it's going to be produced in the sites underneath the traumatic injury. Fortunately, if we want to now stop the production, all we have to do is remove minocycline. At that point, the tetracycline activator will disengage from the episome and IL-2 production should cease. So that's the theory. This is how it works in practice. We're looking at the regulatory T cell numbers in the spleen. Here we have our control vector. Here we have our regular vector without the inducer. And then finally in black, we have the IL-2 delivery system with the inducer of minocycline. In the spleen, nothing happens. We don't get any increase in Tregs. In the brain, we have a clear induction of regulatory T cells after providing minocycline. And if we withdraw minocycline, then we have a reduction back to baseline levels in the brain. So now we can go back to our controlled cortical impact preclinical model of traumatic brain injury, and we can now treat post-impact. So here we now induce traumatic brain injury in the mouse. We're coming after the injury, same as we would in a clinical setting, and giving an intravenous injection of the delivery system. And then we give oral minocycline to activate the promoter and drive IL-2 production. We can then look 2 weeks later at the lesion size, and we can see the lesion size is reduced by about 50% by this treatment. So it does look like this system will work in a more clinical system, a more clinically orientated system of a post-injury treatment and using a safety switch that makes it more appropriate for clinical use. So to summarize this part of the talk, what we've shown is that regulatory T cells in the brain are really limited in number because of the very, very low levels of IL-2 that are present. However, if we were able to go to a patient and inject those patients with an AAV gene delivery system, we can increase the amount of IL-2. The AAV can be delivered intravenously. It's going to go through the bloodstream, and because of the neurotrophic capsid, it can bind and cross the blood-brain barrier. And now, once it's inside the brain, we're going to get the transduction of reactive astrocytes. Those reactive astrocytes that have got incorporated the episome from the AAV, they're going to start producing interleukin-2. And now the regulatory T cells coming to the brain will have enough interleukin-2 so that they can survive, they can settle in the brain for a longer period of time, and they can create this more anti-inflammatory environment that can protect against neuroinflammatory damage. And the use of a oral minocycline trigger for this really allows, I think, translation into the clinic, because once we've finished with the treatment, we can really just stop providing minocycline and the system should return to normal. With that, I'd like to finish up. This has really been a labor of love for a lot of people for a very, very long time. And I want to call out a couple of people in particular, but really the whole team has been amazing. But Lydia Yishi really led on the AAV IL-2 work. Manu Prosciutto really led on the development in the MHC class 2 knockout mice. Pascal Bielefeld is our traumatic brain injury expert. James Dooley has really just been pivotal to the whole project. Matthew Holt, our key collaborator on AAV systems and astrocyte transduction, and really a whole host of very skilled people in our lab and collaborating labs that have allowed us to tap into so many different models and cell biologies. Thank you very much.
I thank you very much. This was fantastic. This was a great talk. Thanks so much for this. And I have a ton of questions. However, this is not the format we have here today. The format is that because there's so many people listening to you right now live, and there will be plenty of people listening to your YouTube presentation, your presentation on YouTube, we asked you to ask Adrian questions via Twitter. What you need to do is you could search for the account Global IMMUNOTALKS and then find the tweet that says ask questions for Dr. Adrian Liston here. And then you'll be able to communicate with Adrian, ask your question, anything you always wanted to know. This is a good opportunity. And I know Adrian is actually going to answer every single one of them. Knowing him, he'll take care of that. So without further ado, this was a fantastic seminar. Thank you very much for this, and I hope to see you in person soon. Thank you, everyone, and good night, good afternoon, good morning. Bye-bye.
Thank you.